4 billion years ago, Earth did not look like this. No flowers, no birds, no quiet mornings with sunlight through a window, just rock, fire, and a sky thick with volcanic gases. Asteroids fell. Oceans may have boiled. The surface was unstable, violent, unfinished, and yet, almost as soon as this planet could hold stable liquid water, life appears in the record. Not late, early, shockingly early, which means one of two things. Either life forms easily when the conditions are right, or something extraordinary happened here. Every living thing on Earth shares the same basic chemistry. DNA stores information, RNA helps translate it, proteins do the work, ATP moves energy inside cells. Very different organisms, one chemical foundation. That unity tells us something profound. Whatever started life on this planet started at once. The scientific question behind all of this is called abiogenesis. It simply means the origin of life from non-living matter. Scientists have proposed multiple explanations. Lightning in a primitive atmosphere, chemical reactions at deep-sea vents, sun-warmed tidal pools, mineral surfaces, even organic molecules delivered from space. Each theory tries to explain the same impossible transition. How does chemistry begin to replicate itself? Because once something can make copies, evolution takes over, and complexity becomes inevitable. We do not have a final answer, but we do have evidence, experiments, competing ideas, and a growing understanding of just how thin the line is between chemistry and life. Tonight, we walk through what we know, what we don't know, and maybe what we might have missed, because the moment matter began to remember itself might be the most important moment in Earth's history. The first serious scientific idea about life's origin is often called the primordial soup. It is simple, and for decades it shaped the entire conversation. The early Earth likely had an atmosphere very different from today. No oxygen to breathe. Instead, gases like carbon dioxide, nitrogen, methane, ammonia, and water vapor, and a reactive sky, full of energy. Add lightning, add ultraviolet radiation from the sun, add constant volcanic heat, and suddenly, simple gases begin reacting. Organic molecules form. Organic does not mean alive. It simply means carbon-based. The chemistry life is built from. Among the molecules produced, amino acids. The building blocks of proteins. In 1953, two scientists tested this idea directly. Stanley Miller and Harold Urey built a glass apparatus to simulate early Earth's atmosphere. They filled it with simple gases. They ran electric sparks through it to mimic lightning. After a week, the liquid inside turned pink, and when they analyzed it, they found amino acids. Life's building blocks had formed spontaneously, from gas, from energy, from chemistry alone. That experiment did not create life, but it proved something critical. The molecules required for life can form naturally under plausible early Earth conditions. Over millions of years, these molecules could have accumulated in the oceans. A chemical broth, a global laboratory, a primordial soup. But here is what we do not know. Making amino acids is not the same as making life. The same way a pile of bricks is not a house. How do you go from simple building blocks to long chains like proteins, to something capable of replication? Water can also break complex molecules apart. The ocean is generous. It is also disruptive. Modern research suggests Earth's early atmosphere may not have been as chemically reducing as Miller and Urey assumed, meaning fewer reactive gases like methane and ammonia. That alone doesn't invalidate the idea, but it complicates it. Scientists have since repeated and modified the experiment many times, using different gas mixtures, different energy sources, meteorite impacts, even volcanic plumes, and organic molecules still form. So here is where the primordial soup hypothesis stands today. We know organic building blocks can form naturally. We do not yet know how those building blocks assembled into a self-replicating system. The soup may have provided the ingredients, but it does not yet explain the recipe. So some scientists looked elsewhere for the missing step, not at the surface, but deep below it. If life did not begin in a sunlit surface ocean, maybe it began in the dark, far below the waves, where sunlight never reaches. The ocean floor splits apart. Seawater seeps into cracks in the crust, heats near magma, and erupts back out through hydrothermal vents. These vents release superheated, mineral-rich water into the cold ocean, and where hot and cold meet, chemistry becomes intense. This idea suggests life began not from lightning, but from chemical energy. When two different chemical environments meet, they create what scientists call a gradient. A gradient is simply a difference in temperature, in acidity, in chemical concentration. Nature tends to push those differences toward balance, and that movement releases energy. Some hydrothermal vents form chimney-like structures made of iron and sulfur minerals. These minerals can act as catalysts, substances that speed up chemical reactions without being consumed. In laboratory experiments, similar minerals have helped produce organic molecules and drive reactions that resemble early metabolism. Metabolism means using energy to power chemical processes. And here's the intriguing part. Modern cells still rely on chemical gradients across membranes to produce energy. Inside every living cell, tiny molecular machines use proton gradients to generate ATP. Some scientists believe hydrothermal vents provided a natural version of that system before cells even existed. Rock first, biology later. Today, entire ecosystems thrive around hydrothermal vents without sunlight. Life powered entirely by chemistry. That makes the idea plausible. But there are still open questions. Can complex molecules form and remain stable at those temperatures? Can self-replicating systems emerge in such turbulent environments? The vent hypothesis solves one problem beautifully. It explains a steady, long-lasting energy source. What it has not yet fully demonstrated is the leap from chemical reactions to true replication. Still, many researchers consider hydrothermal vents one of the strongest contenders, because life, even today, runs on gradients, and the deep ocean had them in abundance. But some scientists think we've been looking at the wrong kind of vent, not the violent black smokers, something quieter, more structured. Not all hydrothermal vents are violent, superheated chimneys. Some are cooler, alkaline, and full of tiny, naturally forming compartments. These structures look less like smokestacks and more like stone honeycombs, filled with microscopic pores and natural chambers. Inside these vents, alkaline fluid meets the more acidic early ocean. That difference creates a powerful chemical gradient, a difference in proton concentration. Modern cells use proton gradients across membranes to generate ATP, their energy currency. These vents may have provided that system before membranes existed. In other words, the ocean floor may have built a prototype cell, stone walls instead of membranes, natural gradients instead of molecular pumps. Those mineral pores could trap organic molecules, concentrate them, allow reactions to occur over and over again instead of dispersing into open water. That stability matters. The minerals themselves, often rich in iron and sulfur, can act as catalysts, helping carbon dioxide react with hydrogen to form more complex carbon compounds, step by step. This model connects geology directly to biology. It suggests metabolism, energy-driven chemistry, began inside rock long before free, living cells emerged. Over time, organic membranes may have replaced mineral walls, cells becoming independent, carrying the energy system with them. What makes this idea compelling is how closely it mirrors modern life. Even today, every living cell depends on proton gradients to make ATP, the same fundamental mechanism. But again, there are gaps. We have not yet observed a complete transition from vent chemistry to self-replicating genetic systems. Energy alone is not inheritance. Still, the alkaline vent model may solve one of the biggest puzzles: how to sustain organized chemistry long enough for something like replication to emerge. Because at some point, chemistry needed to do more than react. it needed to copy. At some point, chemistry had to do something new. It had to replicate. Because once something can make copies of itself, evolution begins. The RNA world hypothesis proposes that before DNA, before proteins, there was RNA. RNA is a molecule similar to DNA, but simpler. It can store information in its sequence of chemical letters. Those letters, abbreviated as A, U, C, and G, can pair in predictable ways, which means an RNA strand can serve as a template to build another strand. In theory, it can copy itself. But RNA can also do something remarkable. Certain RNA molecules act as catalysts. They speed up chemical reactions. These catalytic RNAs are called ribozymes. That discovery changed everything. It meant one molecule could both store information and perform chemical work, a single system doing two essential jobs of life. If early RNA molecules could copy themselves, even imperfectly, natural selection could begin. Variations that copied more efficiently would become more common. Complexity would follow. Experiments have shown that RNA building blocks can form under plausible early Earth conditions. Scientists have also created ribozymes in laboratories that replicate short RNA sequences. Not perfectly, not indefinitely, but enough to suggest the concept works. The challenge is scale. RNA is fragile. It breaks down in water, and assembling long, stable strands without enzymes is difficult. Some researchers propose that RNA formed inside primitive membranes, or on mineral surfaces, or in environments with wet-dry cycles that encouraged strand formation. The RNA world may not stand alone. It may have required help. Still, the RNA hypothesis answers one critical question better than most others. How did inheritance begin? How did information persist long enough for evolution to take hold? If this theory is correct, life began not as a cell, but as a molecule that learned to copy itself. And once copying begins, biology is no longer inevitable, but it becomes possible. But some scientists believe we still have the order wrong. Maybe replication did not come first. Maybe something else was already running. The RNA world says life began when a molecule learned to copy itself. The metabolism-first hypothesis says copying came later. Energy came first. In this model, early Earth's mineral surfaces, especially those rich in iron and sulfur, drove self-sustaining chemical reactions, not random reactions, networks. A reaction network is a chain of chemical steps that feed into one another. One reaction produces a molecule that fuels the next. Energy flows through the system, like a loop that keeps turning. The iron-sulfur world hypothesis proposes that minerals acted as primitive catalysts. They helped carbon dioxide and hydrogen react into more complex organic molecules step by step without genes. The key idea is that life is not defined first by information. It is defined by metabolism, by organized energy flow. A living system maintains itself by constantly processing energy. If early Earth hosted stable, repeating chemical cycles that sustain themselves, that might qualify as proto-life. Not alive as we know it, but not inert either. Over time, those networks could have grown more complex, eventually incorporating molecules capable of storing information, genes emerging inside a system that was already running. In this view, replication did not spark life. Replication was absorbed into an already active chemical engine. The strength of this idea is stability. Metabolic reactions powered by geological energy sources could persist for long periods, long enough for complexity to build. The weakness? No one has yet demonstrated a fully self-sustaining metabolic network that transitions naturally into a genetic system. We can model pieces, not the full bridge. Still, this hypothesis reminds us of something important. Life is not just information, it is chemistry in motion. And perhaps motion came first. Which brings us to another possibility. What if the first step was not genes and not metabolism, but boundaries? Every living cell on Earth has one thing in common: a membrane. a boundary between inside and outside. Certain molecules, called lipids, behave in a very particular way in water. One end is attracted to water, the other repels it. When placed in water, they spontaneously arrange themselves into spheres. The water-loving ends face outward, the water-avoiding ends tuck inside. The result is a hollow bubble. These bubbles are called vesicles. They form naturally. No enzymes required, no biology needed, just chemistry obeying physical rules. The lipid world hypothesis suggests that these primitive membranes came first. Before genes, before complex metabolism, containers existed, and inside them, chemistry became concentrated. In open ocean, molecules drift apart. Inside a vesicle, they collide again and again. Concentration increases. Reactions become more likely. Order begins to emerge. A membrane creates selection. If a vesicle grows and divides, whatever chemistry is happening inside gets carried forward, inheritance without genes. Experiments have shown that simple fatty acids, molecules that could plausibly form on early Earth, self-assemble into stable vesicles. Some even grow and divide under changing conditions. Add internal chemistry, perhaps RNA, perhaps metabolic reactions, and now you have a system that can evolve. A boundary makes Darwin possible. The lipid world does not claim membranes alone were alive. It suggests that life required compartments, a protected interior where complexity could build without being diluted. The challenge is integration. How do membranes, metabolism, and genetic molecules merge into one unified system? We can build pieces in laboratories. We have not yet recreated the full transition. Still, this idea adds something essential. Life is not just chemistry. It is chemistry enclosed. And some scientists believe those enclosures did not form in the deep ocean at all. They formed under the sun, in places that dried and filled and dried again. In a letter written in the 19th century, Charles Darwin imagined life beginning in a warm little pond. Not an ocean, a puddle. Modern science has refined that image into what is called the wet-dry cycle hypothesis. Shallow pools that repeatedly filled with water, then dried in the sun, over and over. When water evaporates, dissolved molecules become concentrated. The more concentrated they are, the more often they collide. The more often they collide, the more likely they bond. This process highlights the cyclical nature of these environments and the physical drivers of molecular concentration. Laboratory experiments show that cycles of hydration and dehydration can help form long chains of molecules, including RNA-like polymers. Water helps molecules move. Drying helps them connect. In constant open ocean, complex molecules can break apart as quickly as they form. But in a pond that dries and refills, chemistry gets rhythm. Concentration, release, and concentration again. Dry phases also expose mineral surfaces. Clay and other minerals can help align molecules, making reactions more likely. The pond becomes a laboratory. Fatty molecules form membranes more easily in these fluctuating conditions. RNA strands may form and become trapped inside vesicles. Now replication, metabolism, and boundaries begin to intersect. This hypothesis has strength because it solves a chemical problem. It explains how long chains, the kind needed for genetic molecules, might form without enzymes by cycling between wet and dry. Its weakness is geological uncertainty. Were such ponds stable and common enough on early Earth? We do not know. But the chemistry works. Life, in this view, may not have begun in the vastness of the ocean, but in small, temporary pools that appeared and disappeared with the weather. And whether in ponds or deep-sea vents, another possibility keeps surfacing. That rock itself may have organized the first steps. What if the first step toward life was not in water at all, but on rock? Certain minerals, especially clays, have highly ordered crystal structures. They are not random. They form repeating patterns at the microscopic level. When organic molecules settle onto these surfaces, they can line up along those patterns. Alignment matters. In open water, molecules drift aimlessly. On a structured surface, they can organize. Laboratory experiments show that clay minerals can help small molecules assemble into longer chains, including RNA-like polymers. The surface holds them in place long enough for bonds to form. This idea suggests rock acted as scaffolding, a temporary support system that guided chemistry toward complexity, not alive, but not passive either. Some minerals can also act as catalysts, accelerating reactions. Iron, nickel, and other metals embedded in rock may have played an active role in forming organic compounds, the planet itself participating. The strength of this hypothesis is control. Surfaces provide order, they reduce randomness, they increase the chances of meaningful chemical connections. Eventually, molecules that formed on rock could detach, drift into water, become enclosed in membranes. The scaffolding no longer needed. The weakness? No mineral surface alone has been shown to produce a fully self-replicating system. We see pieces. We do not yet see the full chain. Still, this hypothesis reminds us of something fundamental. Life did not arise separate from the planet. It emerged from it. From water, from heat, from rock, or did it? There is one final possibility, that some of the chemistry we've been talking about did not start here at all. During Earth's first few 100 million years, it was not alone in its violence. Asteroids and comets struck the planet constantly. For a long time, those impacts were seen only as destructive. But modern research suggests they may also have delivered something essential: organic chemistry. Meteorites that have fallen to Earth contain amino acids, sugars, nitrogen-containing bases similar to those found in RNA and DNA, and complex carbon compounds formed in space. Comets, made of ice and dust, can preserve organic molecules formed in interstellar clouds. Simulations show that radiation acting on simple ices in space can produce surprisingly complex chemistry. This idea is called panspermia. It does not necessarily claim that fully formed life arrived here. More often, it suggests that the building blocks of life were delivered. If early Earth received a steady rain of organic molecules from space, that would have dramatically enriched the chemical inventory available for life to begin. If there were more ingredients, there were more possibilities. Some researchers have even tested whether microbes could survive the shock of impact or the vacuum of space. Certain hardy organisms can endure extreme radiation and cold for limited periods, but survival over interstellar distances remains unproven. The strength of this hypothesis is evidence. We know organic molecules exist beyond Earth. We have held them in our hands. The limitation is deeper. Even if life's chemistry came from space, it still had to begin somewhere. Panspermia shifts the location. It does not solve the origin, but it changes the scale of the question. If the ingredients for life are common in space, then perhaps life is not rare chemistry. Perhaps it is common potential. Or perhaps Earth was simply the place where those ingredients finally assembled. 9 theories. 9 serious attempts to answer one question. How did life begin on this planet? We have chemistry that works in laboratories. We have meteorites filled with organic molecules. We have hydrothermal systems that mirror the energy systems inside modern cells. What we do not have is the moment. No fossil of the first replicating molecule. No recording of the first membrane closing around chemistry. No witness. Every theory explains part of the puzzle. None explains all of it, and that may be the most honest thing we can say. It is possible life did not begin from one process. It may have required many: surface chemistry organizing molecules, wet-dry cycles forming chains, minerals catalyzing reactions, vents providing energy, membranes forming compartments, and perhaps ingredients delivered from space increased the odds, not one theory winning, but several cooperating. The point of this channel has never been certainty. It has been clarity. What we know, what we do not know, and what we might be overlooking. We know that life appeared on Earth at least 3.5 to 3.8 billion years ago. We know it happened relatively quickly after the planet cooled. That alone tells us something important. Either the transition from chemistry to biology is extraordinarily rare, or under the right conditions, it may be almost inevitable. And if it is inevitable, then the question is no longer only how life began here. It becomes how often it begins elsewhere. For now, we continue asking. We continue testing, we continue refining. Because the origin of life is not just a story about the past, it is a story about possibility. For more about the origins of life, please like, subscribe, and ring the bell. This has been For What It's Worth.